Assalamu alaikum. Um, I'm re-recording this part of the lecture because I think that I made a mistake and I better correct it. So راح أعيد التسجيل أوكي علشان في غلطة عملتها في واحدة من السلايدز فبدنا يعني نصلحها عشان نلم الموضوع هيك يكون كله صح. We'll talk about the MAP kinase pathway. The MAP stands for mitogen activated pathway. Sometimes you can find it written as mitogen activated protein. So mitogen is basically a small protein or peptide, and what it does is that it stimulates, it induces cell division or cell proliferation. So we'll start with RAS. RAS is a small monomeric G protein, again meaning that it gets activated when it binds to GTP. And it can activate other proteins downstream. Downstream. Now, downstream means something that comes after something else. Okay, so it's like going downstream. Stream يعني مجرى مي. So ورايح لتحت. فإحنا عمالنا بنمشي لتحت. So RAS is activated when it binds to GTP, and this binding is facilitated طيب بصير أسهل in the presence of GEF proteins اللي هم guanine nucleotide exchange factors. When GTP is bound to RAS, RAS becomes active and it activates downstream effectors, molecules. And RAS is inactivated when GTP is hydrolyzed into GDP. And this hydrolysis is facilitated by proteins known as GTPase activating proteins or GAPs. So the GTPase activity belongs to RAS, جزء من RAS, intrinsic, إشي داخلي خاص the RAS protein. والGAPs, all they do is they facilitate the intrinsic activity. Okay? بيحفزوا الintrinsic activity مش إنه هم اللي بيعملوا الهيدروليسيس. هلا RAS is an oncogene. onco يعني tumor. oncogene هو الجين اللي بسبب التومر وكانسر لما يصير له mutation. When it comes mutated, it causes cancer. Otherwise, it's called a proto-oncogene. So proto-oncogene is a normal gene. It's not mutated, it's under control, it's under regulation, it doesn't cause cancer. Except when it gets mutated, it causes cancer. هلا هذا الرأس موجود بعدد كبير من الكانسرز like pancreatic cancer. 95% of pancreatic cancers have mutated RAS, oncogenic RAS. Colorectal cancer also has a good percentage of colorectal cancer also has mutated RAS as well. So the way it works when RAS is activated is the following. Okay, let's go to the next slide. So the ligand binds to the receptor. The receptor dimerizes. Then you have autophosphorylation. So you have this domain phosphorylating this domain and this domain phosphorylating the other domain. Autophosphorylation. Even though these are different proteins, we call the process autophosphorylation because they are the same proteins. So the phosphotyrosines create a docking site, a site whereby other proteins can bind to the receptor. So we have an adapter protein that binds to a GIF or GIF, a guanylynucleotide exchange factor that helps RAS replace GDP for GTP. So GTP comes in, GDP comes out. Actually, it's GDP comes out, then GTP comes in. طيب، فالجوانينيوكليوتيد اكسشينج فاكتور لما يرتبط مع الكاينيز دومين عن طريق الادابتر بروتين بحفز أو بسهل عملية الاكسشينج. Note that RAS is bound to the plasma membrane. So all that happens is that these proteins come closer to RAS. And this is the whole purpose, is to put all proteins in close together in one place. Now, RAS binds to a kinase, an effector molecule called RAF. RAF gets activated when it binds to RAS, and RAF activates another effector molecule. So we have amplification happening. This kinase molecule called MEK phosphorylates another kinase, another amplification called ERK. Now ERK goes into the nucleus and it phosphorylates and activates certain transcription factors. فالعملية إن RAS becomes active. RAS بيعمل activation لRAF. RAF is a kinase بيعمل phosphorylation لMEK. MEK هلا صار active. MEK بيعمل phosphorylation لإرك. إرك صار active. إرك بدخل على النوكليوس وبيعمل phosphorylation للtranscription factors. هاي transcription factors activate the expression of genes that promote cell division and proliferation. فلما يكون الRAS mutated بمعنى إنه it's always active, RAF is always active, and this pathway is always active, and the cells are always dividing. And that's why RAS is an oncogene لأنه it causes cancer. So, another pathway is the PI3 kinase AKT pathway. هذه pathway is a pro-survival pathway. A pro-survival pathway. And what happens you have the ligand binding to the receptor is activated, it dimerizes, you have autophosphorylation creating a docking site. حكينا إنه docking يعني زي إنه السفينة لما توصل على الميناء وتدخل في المكان تبعها المرساة، هذا الدوكينج سايت، المكان اللي بتنربط فيه السفينة أو القارب اسمه docking site. So what binds to the receptor, to the phosphorylated receptor, is the enzyme PI3 kinase. PI3 يعني phosphatidylinositide. هلا phosphatidylinositide it's a phospholipid. Okay. هذا زي الفوسفاتيديلينوسيتول. اوكي. بس عليه فوسفيت جروب. هلا هذا what the enzyme does انه it converts PIP2 to PIP3. PIP2 is phosphatidylinositol 4,5 bisphosphate. بصير phosphatidylinositol 3,4,5 triphosphate. هذا موجود في البلازما membrane. This exists in the plasma membrane, and it becomes the binding site. PIP3 becomes a binding site to 3 signaling molecules. الثلاث هم AKT, mTOR, and PDK1. اوكي ماشي، نروح على السلايد اللي بعديها ونكمل. So ligand binds to the receptor. receptor dimerizes. It gets phosphorylated, autophosphorylation, creating a docking site for PI3 kinase. PI3 kinase converts PIP2 to PIP3. And um, um, PIP3 is the binding molecule for three signaling molecules. AKT is one of them. The other two are mTOR and PDK1. When they get close together, when they get nearby each other, بيقربوا على بعض. فwhat PDK is a kinase, mTOR is also a kinase, and what they do is that they phosphorylate AKT. AKT, when it gets phosphorylated, it becomes active. Okay? Now, what it does is that it inhibits cell death, so promoting cell survival. It controls a number of transcription factors, and what it does is that it activates translation of proteins. لأنه هذا GSK is an inhibitor of translation. هذا GSK gets inactivated by AKT. فبالتالي AKT activates translation. So it is a pro-survival molecule. أو signaling pathway is pro-survival. This shows PIP2 being converted to PIP3. Okay? All of these signaling pathways work in harmony together. They do not do their work by themselves. There is crosstalk, there is interaction between the different signaling pathways. So I'll give you some examples of how these pathways work together. Well, first we'll talk about feedback loops and how they work. So remember in biochemistry how we talked about feedback regulation. So for example, in glycolysis, you have the phosphorylation of glucose, then to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 1,6-bisphosphate, etc. Now what happens is that if there is a lot of glucose 6-phosphate, it's going to inhibit the hexokinase. If there's a lot of fructose 1,6-bisphosphate, it's going to inhibit the phosphofructokinase, and so on. If there is a lot of ATP, it's going to inhibit the glycolytic pathway, because there's no need to degrade or break down more glucose. Let's store it instead. So same idea happens in signaling pathways. An example is a protein known as NF-kappa B, nuclear factor kappa B. Now, this protein, when it's free, when it's active, that is, it goes into the nucleus and it induces the expression of several genes that induce mutation. And this is an important pathway in the immune system. One of these genes is known as I-kappa B. This is an inhibitor of NF-kappa B. It's an inhibitor of this molecule. It binds to it and it prevents it from getting into the nucleus. Okay, so this is feedback mechanism. Now, so what happens is that if you have a signaling pathway that is activated, what it does is that it phosphorylates the inhibitor of the NF-kappa B, releasing it from NF-kappa B, and it gets degraded. Now NF-kappa B becomes free, so it goes into the nucleus, again inducing the expression of several genes, including I-kappa B. Again, it inhibits NF-kappa B from getting in. So the cycle goes on and on, balancing out the expression or the activation of this pathway. Another example of signaling homeostasis and balancing things out is the nerve growth factor molecule. Now, it binds to its receptor, activating the RAS pathway and the PI3 kinase pathway, the pro-survival pathway. Now, the RAS pathway induces proliferation. Now, if the signaling pathway is activated for 30 to 60 minutes, you have the activation of the RAS pathway, but not the PI3 kinase pathway. Now, RAS induces cell proliferation, but if the signal prolongs, if it becomes active for a longer time, then the other pathway, the PI3 kinase pathway, is activated, not the RAS pathway. And this pathway induces differentiation of cells into neurons. So again, initially you have activation of cell proliferation by activating the nerve growth factor signaling pathway. But when the signal continues to be active for a long time, then you have activation of the PI3 kinase pathway that induces cell differentiation. So again, here we have regulation, nice regulation, signaling homeostasis. The third example to show you how signaling pathways can be regulated is crosstalk of signaling pathways. So we have two pathways again. We have the RAS pathway and we have the PI3 kinase pathway. When you have activation of the receptor by its ligand, you have activation of the RAS pathway and activating RAF. RAF activates the PI3 kinase, and PI3 kinase can then activate AKT. AKT in turn inhibits RAF. So there is a limit by which RAF would be activated for. So it doesn't go out of control. It doesn't get active without any inhibition. Rather, you have this pathway that inhibits the RAF molecule. Now, eventually, anyhow, if RAF is active, it activates MEK, like I said before. MEK activates ERK, and ERK activates the downstream molecule down there, the mTOR, which activates, by the way, the AKT as well. So you have this complex interactions of the different molecules, but the whole purpose of this whole thing is to balance things out. So yes, if ERK eventually gets very active, it activates the mTOR that activates AKT. When AKT gets really active, it inhibits the RAF pathway, which eventually will inhibit the ERK, so that ERK doesn't get active all the time. So things get balanced at the end. So the question is now is, how do cells do all of that? Sometimes cells can respond to the same hormone, but in different ways. Cells can have the same hormone and the same receptor, but still they respond differently. So how do cells do that? Well, there are different mechanisms. One, you can have cells having different receptors. So you can have the same ligand acting on the same cell, except that it binds to different receptors. Maybe cells have the same receptor, okay, but they behave differently. They respond differently, and the reason is that because they have different effectors, they have different transducers. They respond to secondary messengers in a different way. So, for example, you can have activation of protein kinase A. Protein kinase A would then respond differently because it has different effectors. You can have the same secondary messenger. You can have cyclic AMP, but cyclic AMP binds to different effector molecules. Also, if we assume that the final target is the same, that is DNA, still DNA is not the same in all cells. Yes, they have the same number of genes, but remember, we have heterochromatin and we have euchromatin. Heterochromatin is when DNA is condensed, it's packed, it's not accessible by transcription factors. Euchromatin is when you have the DNA loose, the DNA is accessible for transcription factors. So what happens is that you may have the same signaling pathway, the same receptor, the same effectors, the same secondary messengers, the same transcription factors activated. But when they get activated, they have to bind to DNA. If DNA is a heterochromatin in the cell, it cannot be bound by the transcription factor. It can be a euchromatin in a different cell, and cells would respond. Okay? The gene transcription would be activated because it's euchromatin in that cell. So again, every cell has its own what we call a molecular profile, a molecular fingerprint. So keep that in mind.